I can empathize with our next speaker. His last name starts with a W, and as a result of that, he's going last. And with a name like Zylstra, I'm used to that. And by the way, I was at your facility during Night of the Open Door with my 17-year-old son. It was really a great night. Neal Woodbury is the co-director of the Biodesign Institute and is Innovations in Medicine professor at ASU's College of Liberal Arts and Sciences. He leads a team that seeks to develop molecular devices and nanoscale hybrid electronics for use in biomedicine. Also for use in environmental remediation and monitoring, threat detection, and agriculture. His research into the structure and function relationships and photosynthesis led him to realize the awesome potential of harnessing the energy of light to direct chemical reactions. Dr. Woodbury is an advocate of interdisciplinary science as a means of providing researchers greater vision and addressing real-world problems. He's been a member of the National Science Foundation's Biophysics Panel, is a current member of the NSF Integrative Graduate Engineering and Research Traineeship Program, and has served as the director of the Photosynthesis Center at ASU. Dr. Woodbury received a BS in biochemistry from the University of California at Davis and its PhD from the University of Washington. Dr. Woodbury?
So how many of you have seen this movie? This was a while back, so a few of you have. Probably about half of you weren't even alive when they made this movie, so I'll recap it for you real quick. Basically, they took Raquel Welch and they shrunk her down to something a little smaller than a blood cell, put her inside of a shrunken submarine, and sent her into somebody to fix his brain, okay? Now that, since we were asked to do something unbelievable, is pretty unbelievable. But actually, there are things going on inside of you right now which are probably just as unbelievable. And Stuart actually referred to a number of these things earlier on. This is a picture I stole from Roche, a company that does a lot of diagnostics and other aspects of biomedicine. And what you see here is an artist's rendition of something called a B cell, which is that orange blobby thing there in the lower part. And what that B cell is it's one of these cells that's a bit like that what we envision that submarine to do. It runs around and it basically tries to figure out what's you and what isn't you. And when it finds something that isn't you, it tags it so that we can get rid of it. Now the very cool thing about B cells is that they are basically repositories of a tremendous amount of information about what's going on inside of you. So we have these, they're not quite nano, I'll call them microbots. We have these microbots running around inside of us, which know us in tremendous detail. They learned about us when we were very young, sort of got imprinted with what we were from a molecular, at a molecular level when we were very, very young. And they run around inside of us looking for things that are different. Now those different things could be a virus, you catch a cold, or a bacterium, or a cancer cell. And as we grow older, part of the problem is, for those of us that are growing older realize this, our immune systems don't always recognize us quite in the same way anymore. And they can either have the problem of recognizing things they shouldn't, in which case we get things like rheumatoid arthritis, and these are autoimmune problems, or they don't recognize bad things when they should, in which case we get the cancers that Stuart talked about earlier on. And so it's a very interesting aspect of these things that they're gathering all this information all the time. They have all this information. It becomes, as we grow older, in particular, a decision-making process. They don't do as good a job of making decisions as we get older because although they have information, they don't know exactly what's changed and what hasn't. So why would we be interested in this information? this information is essentially an imprint of what's happening in your body all the time. So if we could access that information, we would know a tremendous amount about it. You've got about 1 billion of these different B cells running around inside of you all the time, looking for things and storing that kind of information. One of the reasons why we'd be very interested in knowing that information is exactly what Stuart mentioned in his talk, and that is that we would really like to put cancer in the rearview mirror, or at least get it to a point, as he said, where it was manageable. The best way to get it to a point where it's manageable is to know about it a lot earlier. And some of the statistics that I stole from various websites up there are shown. And when you look at that, you'll see that basically if we know about cancer early on, we can usually fix it, or at least manage it. If we don't know about cancer until it's in a late stage, then it becomes much, much more difficult to manage in an appropriate way. So I'm going to tell you very quickly about a technology that a couple of my colleagues have developed to try and download that information. Okay, so basically the question is, how do I take that billion-fold set of cells that are going around gathering all that information and download that information into something that I can analyze and understand from a diagnostic point of view or from a medical point of view. So what these two fellows did, Dr. Stephen Johnston and Dr. Phil Stafford, was they reasoned that if they put stuff down on a surface that was sufficiently complex, that these things that are always looking for different things, that's what these cells do, and what the antibodies that they create do, well, the antibodies would bind in different places on the surface and they'd get a picture, okay? And so they did this, and they created a surface with lots and lots of little pieces of protein on it, is what they have. And by lots and lots, I mean thousands. Actually, the current versions are hundreds of thousands of these little pieces of protein on a surface. And what happens then is that the antibodies that are circulating around in your blood will bind to that large array of these little biosensors. And what you'll get is a picture. And that picture changes depending on the disease. So if you have breast cancer, you get one picture. If you have the flu, you get another picture. If you have Alzheimer's, you get yet another picture. And so this picture then is extremely useful in terms of understanding the way disease works. Now, how this happens is it only requires a very, very small amount of blood. It takes actually only something called a microliter, which is about a 20th of a drop of blood. And you can actually dry it on a piece of filter paper. You could, once this is hopefully about 1.5 years from now, this will become available, you will be able to put this on a piece of filter paper and actually even send it through the mail. And the small amount of blood then is diluted and put on one of these surfaces. And what these surfaces actually look like is this. It's probably a little hard to see in the light, but maybe you can see some sort of a slightly colorful picture here. This is a slide. It's a piece of a silicon wafer. All of these molecules are built on silicon wafers, just as I build the things that are in my cell phone on a silicon wafer. Same process, same type of lithography and whatnot that's used to build that is built this. And so they're built on this surface, and actually this is kind of, see if I can get this to work in here. I made a nice dark background. I'll see if I can bounce this off of here. But there's so many of them on here that I can actually, maybe not. No, not really. Well, sort of. Okay, not very good. But if you use your imagination, you can see one dot with a bunch of little dots around it. And what's happening there is that this is an array of these little biosensors, and that array of little biosensors is actually diffracting light in such a way that it's showing you the shapes of those biosensors. It turns out they're all shaped in that shape. They're in an orange crate type of arrangement. And each of this square here has 24 different assays on it. Each of those assays is 350,000 of these little biosensors. And all of this is just built in the same kind of equipment that we build your electronics for your cell phone or something like that at that kind of resolution. They're about 8-micron little biosensors. So there's now a company which is starting to build these things. And that company hopes to have these things out in just about 1.5 years, something like that. So basically, the concept here is, can we actually take this incredible information storage that exists at all times, constantly monitoring your body, and download it into some kind of digital form that in fact we can pull out and use. And furthermore, the idea behind this particular technology is, can we do this extremely inexpensively? So this is now something that, like the rest of the electronics industry, gets cheaper and cheaper and cheaper as we shrink it down farther and farther and farther and make more and more and more of them on a single wafer. We're hoping this will come out at a little over $100 an assay to begin with, and then go down from there, with the idea being that it can be used not only here but ultimately throughout the world. So hopefully that's in the not too distant future, and hopefully at that point at least we'll start to put some of those diseases in the rearview mirror. Thank you.